Now let's talk about the morphology of H. pylori bacterium. See, as I said, that this bacterium, it is a motile bacterium. Okay, motile bacterium. Motile bacterium means it have certain structure which help in the motility, and that is one of the strongest, you can say, mostly they are present in the H. pylori bacterium. Okay, and this is called as the flagella. Okay, about this Helicobacter flagella, why I am saying it is a unique, just because it shows the most motility. Okay, it's a you can say a powerful, a viable bacteria, just because due to presence of this flagella, Fine. Now, the another thing about this bacteria is that look at their shape. As I said, the name which is governed is just because of, you can say, their shape. Their shape is like helical shape. That's why they are known as a Helicobacter. Fine, no problem. But their shape can vary. When it goes to the inactive state, it still become the coccus shape. Okay, referred as the coccoid shape. Fine, no problem. Now, so about their morphology, if you forget everything, you have to remember 2-3 things which is very much relevant if we talk about the H. pylori bacteria. The first thing which you should remember is that their shape is the helical shape. Got the point? No problem. And another thing is that it have the more powerful flagella as compared to the another bacteria. Under gram-negative gastrointestinal rod, it possesses one of the powerful flagella. Okay, it possesses one of the powerful flagella. Fine, no problem. Now, so let's see their morphology in our regular, you can say, the headings. Fine. So first is their general morphology. If we talked about their shape, it's a helical shape. But in inactive state, remember, in inactive state, it become the coccoid shape. Okay, it's become the coccoid shape. Fine, no problem. Now, talking about their length varies, okay, between 2.8 to 3.3 millimeter. A micrometer. Okay, it's a micrometer. Now, if we talked about their motility is rapid, remember, and corkscrew movement and rapid movement. Fine, no problem. Now, if we're talking about their lab characteristics, their clinical morphology, it is a fastidious microorganism. Now, what I mean by the fastidious microorganism? Fastidious microorganism refer that it can grow in a wide range of nutrients. It can grow in the presence of wide range of nutrients. Fine, and bacteria love to have chocolates. Yeah, as humans. So same thing is here also. It's grow on the chocolate agar. But it's not exactly a chocolate. It's appear as a chocolate color. Fine. Now, their requirement is we have to, if in order to see this bacteria, in order to, you can say, nurture this bacteria, we have to put the 5 to 20% carbon dioxide. Okay. And you can say most optimum temperature is the 37 degrees Celsius, and about, you can say, their pH, they are basically a acidophilic bacteria. Okay, remember, they are acidophilic. Therefore, it's love to have the acidic environment. Fine. And by providing all these things, it will grow. Okay, it will grow to 2 to 7 days. It will grow on the colony plate 2 to 7 days. Fine, no problem. Now, let's talk about their antigenic morphology. As I said, that this flagella is responsible for causing those diseases related with the H. pylori. So they have 2 type of flagella, remember. The one is called flagella A type and flagella B type. Fine. This both the type of flagella, they will help in the motility first of all. Another thing is in the colonization. Okay, first is the motility, another is the colonization, and apart from that, it's also help in providing the virulence, providing the capacity to cause the disease in the humans. Fine, no problem. Now, another thing is that it have urease enzyme. Okay, put the stark mark over here. This enzyme, okay, this enzyme, it break down the urea into the two things. Okay, this enzyme, it's break down the urea, it's break down the urea into the ammonia and carbon dioxide, and thereby it neutralizes, the hydrochloric acid. Fine. The next thing, which is a more dangerous things about this bacteria, is that it have one gene called as the CAG gene, CAG-A gene. Okay, the full name is cytotoxin associated gene A. Let me repeat it once again. It's worth to repeat it. It's called the cytotoxin associated gene A, and this gene is a carcinogenic gene. It's a carcinogenic gene. Fine, no problem. Now, talking about all these related their morphology, the things which you should remember is that this bacterium, we talked about this bacterium morphology, then remember their morphology is like three morphology. The first one is a general morphology, shape as appear, it's a helical shape. Okay, and in inactive form it's become the coccoid shape. And if we talk about their length varies 2.8 to 3.3 micrometer. No problem. And it's a motile bacteria, and the motility is rapid just because of presence of unipolar flagella. Okay, their clinical morphology, remember this bacteria love to have the chocolate, so it will grow on the chocolate agar, and the temperature is 37 degrees. Temperature is optimum. 6 to 6 point, you can say 3 or 7 is the optimum pH, and they are acidophilic. Okay, and they will grow all about the 2 to 7 days. Fine. Now, the main thing about this H. pylori bacterium is that about their antigenic morphology. Their antigenic morphology is flagella FLA, FLB. These are two type of flagella, and both the flagella is you can say are dangerous. Okay, both the flagella is dangerous, and they help in the motility, they help in the colonization. Okay, and it also help in providing the antigenic property to the bacteria. Urease is the one enzyme which will neutralize the hydrochloric acid bacterium action, just because they will convert the urea into ammonium and carbon dioxide. Fine. Now, another important thing is the CAG-A, and this CAG-A is called cytotoxin associated gene, and this gene is responsible for causing the cancer. So it is all about the morphology. It is all about the morphology of this H. pylori bacterium. Now let's move ahead and discuss their pathogenesis. Now, lab detection, now you should understand. By taking the sample of a stool, okay, if we talk about their lab detection, then you should remember stool examination, okay, and biopsy is the most, providing the most accurate you can say information about the H. pylori. So stool and biopsy test is generally employed to find out the presence or absence of H. pylori bacteria in infected person. Fine. And now this, and if we talked about their transmission, then remember their transmission is from the oral route. Okay. Fecal route as well as sexual route. Okay, so oral route, fecal route, and sexual route is the, you can say, transmission, or you can say the entry area of this bacteria. And remember, that's a human-specific pathogen, so it will transmit from one human to the another humans, one human to another human. Now let's talk about their pathogenesis. Okay, let's talk about the H.pylori pathogenesis. Now, you know that H.pylori bacterium, H.pylori bacterium, it will not respond to the innate immunity. Find that. Now, one of the, you can say, physiological barrier is the hydrochloric acid, isn't it? It's the hydrochloric acid. So it is not responding. It is not responding to the hydrochloric acid. Not responding the hydrochloric acid. Yeah. And you know that it secretes the urease, fine. And basically what happened, let's draw the diagram. See, this is a gastric mucosa. I'm showing the gastric, you can say, epithelial lining. So, what this bacteria does, you know that here is presence of gastric gland. Gastric gland. Now, this bacteria, it's come over here, okay, and it's start penetrating deeper. Okay, it start. First, it causes the inflammation. Inflammation, fine. First, it causes the inflammation, and referred as the very good. Referred as the gastritis. Now, after causing the inflammation, it starts the ulceration. It starts, you can say, decoating the epithelial lining, and that is referred, as the ulceration, or called as the gastric ulcer, peptic gastric or peptic ulcers. Now, this is not limited to only peptic ulcer. It's move ahead and causes, it causes, you can say, first it's knock certain precursor cells. Precursor cell, okay, in the gastric epithelium. Suppose this is gastric, you can say, epithelium nucleus, and this bacteria, it's triggers, and it secretes some of the abnormal protein. And you know, like, whenever the abnormal protein is sensitized, cells loses the capacity, you can say the controlling capacity, fine, and there's a start of atrophy. Okay, write it down. Atrophy of gastric mucosa, fine, and that result into the interstitial metaplasia, a pro form of, you can say, the cancer, and thereby it causes the cancer, or also called as the gastric lymphoma. Gastric lymphoma. Got the points once again? This H.pylori bacterium, they're present in the peptic area of the stomach, we know. And first, its approaches is adhered over the, and you know what? The thing is, it's a very clever. It's a clever. So, it first, you can say, it produces the urease, which neutralizes the HCl and make one cover. You know, see, it's by the synthesizing the urease enzyme, by neutralizing the hydrochloric acid, it's cover one protective layer of urease over the protective film, you can say, over their body. And now it's protected. It's totally protected. So, and the hydrochloric action will not respond here. Hydrochloric action will not respond here. And you know, there is a mucus coating over the gastric epithelium. And mucus generally trap the microorganism. They, you can say, diminish the motility of bacteria. But just because presence of their powerful flagella, it will swim at a faster rate in the mucus. gastric mucus, and it will go to one safer place where there is a minimal action of, you can say, the gastric HCl. So it's learn, it's adapt, is find their natural place, is find their place where it are totally safe. Okay, so it goes to the deeper area where there is no any HCl secretion, and it start causing the inflammation, therefore as a gastritis. Then there is a gastric or peptic ulcer. Then it start disturbing the genetic machinery of the gastric epithelium, and thereby disturb the precursor cells, which result into the atrophy of gastric epithelium, and that result into the metaplasia. Yeah, and that will result into the gastric lymphoma or gastric cancer. Fine. So their histopathology, we talked about the histopathology. Then the initial histopathology is what? It causes the gastritis or inflammation. Okay, gastric inflammation. Fine, no problem. Now, after discussing all these things, let's discuss about the treatment. The treatment, as I said initially, we have to look the population first of all. Is in the endemic see, I will first write down certain antibiotics name. So it's better. One is called clarithromycin, amoxicillin, tetracycline. Okay, and one is not antibiotic; it's a solution called the bismuth. Fine. Now, so initially, this bacteria was bombarded with the clarithromycin antibiotic. Fine, but it seems that these bacteria will develop a resistance toward the clarithromycin. So generally, if a person will not respond, you can say there is a poor prognosis of this disease, or you can say poor prognosis, or there is a less eradication of this bacteria, then we have to go with the amoxicillin and tetracycline. Okay. Meanwhile, you can also administer the bismuth. Bismuth is one, you can see, it provide one protective lining over the GIT mucosa, or you can say it's provide a layer. Okay, and this layer, it is not invaded by the H.pylori. So gastric mucosa become the safe. Okay, gastric mucosa become safe. So hope you clear with this concept. Let's conclude today's lecture. What we studied, we basically discussed about the H.pylori bacteria, a bacterium which is called as the primary carcinogenic bacteria. Fine. In the, you can say, advanced state, it causes the gastric lymphoma. But initially state, it start inflamming, called as gastritis, then peptic ulcer, duodenal ulcer, fine, and then it disturb the precursor cell of the gastric epithelial mucosa, and thereby it causes the, you can say, the cancer, fine. And this H.pylori, Helicobacter pylori bacteria name is decided just because of their present shape. Helical shape, it's a helical shape, and it's present in the pyloric region, so that's why it's called as the Helicobacter pylori. Fine. And their discovery is interesting. Yeah, their discovery is interesting. A scientist drank the fluid from the infected person, okay, and they start testing in himself. Okay, start testing in himself, and thereby it's proved that the peptic ulcer is not, you can say, acquired from the food, or you can say it's acquired from, it's inherited. It's just because due to the H.pylori bacterium. Then we've seen their transmission. It's a human pathogenic bacteria, so it will transmit from human to human through the oral route, fecal route, or it can transmit through the sexual route. Okay, we've seen their morphology. Okay, it grow on the chocolate agar, and the main thing which we have to remember is the antigenic property, antigenic morphology of this bacteria. Okay, they are fragilized antigenic. Apart from that, the second one called proteins, which is a carcinogenic gene called as the CAG gene, cytotoxic associated gene A, which causes the cancer. Then we've seen their treatment. Their treatment is basically, you can say, their treatment is specific to the specific people, because some people have the resistance towards the clarithromycin, so we have to go with amiloxycycline or tetracyclines, okay? And we can also administer one chemical compound called as a bismuth, which will make a film over the gastric mucosa and protect the gastric mucosa. So hope you understood today's lecture. If you have any doubt, comment down, and also if you have any problem related with the handwriting or maybe any sound problem, so you can comment down. We will try to improve. Okay, so thank you for having me, and bye. Have a nice day.